Pedigrees. Before we outline what a pedigree is and complete some associated practice problems with it, I want you to please ensure that you have this note. This was the note that we took in class, and it is imperative that you use this as a cheat sheet as we go along throughout this video and complete the associated questions. If you weren't here to take the note with us, I highly suggest you take this down right now, take a screenshot of it, whatever, and just make sure that you have this at your disposal as we go along in this video. We did go into detail regarding this cheat sheet and what these different components mean and how they can be beneficial to figure out the family tree and to decipher it appropriately. So what is a pedigree? It traces the path of an allele from parents to offspring. So it literally is a family tree. It shows how a trait is transmitted within a family over generations. So the only symbols that you need to be aware of as we go along in this video are the following. So if you have a square, this is representative of a male sex. If it is a circle, it is representative of the female sex. When there is a line that is crossed between one another, this means that they have mated, meaning that they're going to give rise to some children, for example. All right, so this is a female offspring and this is a male offspring. Sometimes twins occur as well. So this triangle, for example, is indicative of that if they are shaded in. Okay, this is representative of an affected disorder. If they are not shaded in, this means that they are not affected. Sometimes a symbol of a dot in the middle could represent carrier. But in most pedigree charts, especially in the course that you're taking, a carrier is not shown with a dot. However, it's just good to know. A slash in between is representative of death. There are other symbols, but these are the only ones that you are likely going to be witnessing throughout this course. An autosomal recessive pedigree is used to represent, disorders that are carried by the autosomes. Remember, autosomes are the body cells. So that is what this is indicative of. But how about this recessive portion? It obviously means that it is inherited as a recessive trait. An important thing to note is, if this is the case, it often skips generations. And I'll show you some examples as we go along to help with your understanding of this, because they are hidden within heterozygous carriers. A good thing for you to note as well is that both parents of an affected individual must be heterozygous. All right, have a look at an example over here. As I mentioned earlier, we do have symbols for carriers. However, we don't always show them, as we want you to be able to analyze the information to determine the answer yourself. So take out that cheat sheet and let's determine if this is autosomal, and let's prove why this chart here is autosomal recessive. So, starting at the top, all affected individuals have an affected parent. Is that true? If yes, it's dominant. All affected individuals have an affected parent. Obviously not true, right? These are not affected. So this is recessive. All right, is it sex-linked? in particular the X-linked, or is this autosomal? Which sex is more affected? Is it mostly affecting the males? If so, it is X-linked. But in this case, it seems as if it is 50-50, so it is going to be autosomal. So now we have to be adding in our alleles. This means that we have to use big R, small r, whatever letters you want to be using. Because if it was X-linked, you would have to be doing XX and XYs along with these ones, right? So something like that, right? But that's obviously not what we're going to be doing. We're just going to be using the autosomes over here. So we're going to be pairing these. If we have a male here and a female here, what do their allele pairings have to be to ensure that the offspring are all unaffected? Well, we know that the female has to be small r, because the disorder is a recessive. And the only way that a recessive can be shown phenotypically is if it was a small r. So then this one here could be a big R small r or a big R. Let's start off with this one. And if it doesn't work out in the future generations, then we can change it to a small r, and maybe that'll work. So let's start with this. As you can see, we have a big R, and a small r. So all of these are going to end up being heterozygous. So there's a 100% chance. And as you can see, all the children over here are big R, small r. So 4 out of 4 being big R, small r is indicative that the parents are both homozygous, as shown over here. Because look what happens if we had done the male as being big R, small r. Then half the chances of the offsprings would be small r, so to be affected. As you can see over here, it's not as if there is a 50-50 chance. It looks pretty much 100% chance healthy. Now, don't get me wrong, it is possible for this parent, this male, to be a big R, small r. Because even though there is a 50% chance of the offspring being unaffected, so normal, it can still result in this, big R, small r, big R, small r, big R, small r, big R, small r. But it is more likely that it is homozygous up here for the parent male, because this is resulting in a 100% genotype of unaffected or normal individuals. So that's what we see here. So we're going to stick with that. So this will be big R, small r, big R, small r, big R, small r, big R, small r. So then what does this one have to be? This male. Well, we know for the female has to be big R, small r, and the male has to be healthy, so it could be big R, or big R, small r. But as you can see here at the very bottom, it is shaded in, which means that this is affected, right? Affected. So there, this needs to be a small r. The only way that could happen is if we keep it the way it is as we see over here, because we're going to be able to get that. Had you had used big R, it would be impossible to get a small r offspring over here. So for sure, this male has to be a big R, small r. And then these individuals over here are unaffected, so normal, so they could be big R, or big R, small r, or this big R, small r as well. In that case, we tend to use R dash and R dash. This means that this could be a big R, or a big R, small r. Same thing with this one. So we have solved it. Let's try another one. Now looking at autosomal dominant pedigrees. This is used to represent disorders carried by the autosomes. Alright, it's the same thing, but the trait is found in every generation. And sometimes the males and females are equal in number of being affected. Alright, let's start again as if we don't know the answer. When do all affected individuals have an affected parent? Yes, these have an affected parent. This has an affected parent, so this is dominant. Alright, is it autosomal or is it X-linked? So let's do the checklist. All affected males have affected moms. Male, affected mom. Male, oh, not affected mom. So this is probably going to be autosomal. How about the next checklist to double check? All affected fathers have affected daughters. All affected fathers have affected daughters. Oh, no, that's not the case over here. So it's leaning even closer to autosomal. And all affected fathers have no affected sons. All affected fathers have no affected sons. Oh, that's obviously not true. Same thing over here. So this is definitely autosomal. So let's make our Punnett squares. And we've got to be using big R's and small r's, right? It's not sex linked, but it's the dominant ones, R, okay, that is affected. And the smaller R is normal, not affected. So this male over here has to be big R, small r. All right, this one here also has to be big R, small r. Why can't I make something like that? Okay, so why can't I do something like this? Why is this one incorrect? Because if you were to do your cross, look over here. Big R, big R, big R, small r, big R, big R, big R, small r. All of them ended up having the disease, being affected. But as you can see over here, that person is not affected. So then that would be a mistake had you gone down that path. So it is for sure like this. So then you have a 25% chance of having a small r, which is normal in this case, right? Because this is a dominant disorder. So you have big R, small r, big R, small r. This one is obviously small r, small r. These ones, however, could be a big R, or a big R, small r. So we'll just put R dash, R dash. All right, how about these parents? Obviously, this female is normal, so I'll be putting a small r over here. What does this male have to be? Well, what if I put a big R, big R? So if I were to do a little Punnett square here again, if I did a big R, and a small r, all of them end up being big R, small r, right? 100% chance. This is a problem because one of the offspring is normal, right? This one has to be a small r. So this is obviously incorrect. So we got to be changing this to a small r. So when you cross these, you end up getting 50% chance of it being small r. This one here, what does it have to be? Well, there is no big R's over here. It's just big R, small r possibility chances. So this is obviously a big R, small r. Now let's do our last one. I don't think we need to do a cross for this one. We have small r, cross with a small r. It's obviously going to be a 100% chance of being small r. So all these individuals are normal. All right, let's try another. So hopefully you get the feel for it now. So now let's look at X-linked pedigrees. This is when the trait skips generations. Also important to note that affected fathers, like I've already said, do not pass to their sons. And here another hint is the males are more often affected than females. Again, this is already in your cheat sheet, right, that I told you to have in your arsenal. So a few examples of these, also obviously indicative for males, is like testicular feminization syndrome or hemophilia, right? We looked at that already in the previous unit. Hemophilia, when you can't stop bleeding, or red-green color blindness, for example. So again, we don't need to really solve this. You already have we've already done two together, that you already have an outlook of how to solve these kind of questions, right? I just want to really show you an example of this pedigree, like a snapshot of it. So as you can see, males are the only ones that are affected. So is this dominant or recessive though? So let's do the checklist. All affected individuals have an affected parent. Affected individuals? Nope, not affected parents. Same thing with this one, not affected parents. So this is probably recessive. So is it affecting mostly males? Absolutely. All the ones that are affected are 100% males. So this is obviously X-linked. All right, how about X-linked dominant pedigrees? For this one, the trait is actually common in pedigrees, and affected fathers always pass the trait to their daughters. And the males and females are equally likely to be affected. Now, these type of diseases or these disorders are actually quite unusual because often they're actually lethal before birth, in males in particular, and therefore only seen in females. So skin lesions is maybe a term that you've heard of before, or rickets. These are indicative of X-linked dominant disorder. So again, another example for you to look at. As you can see, all affected fathers have affected daughters. Here too, right? Affected daughters. Here too, the father, affected daughters. And all affected fathers have no affected sons. All right, son here, son here, son here, son here, son here, not affected. Son here, not affected. Son here and son here, not affected. So Y-linked pedigrees are only found in males, all right? Only found in males. I'll just put in brackets here, never in females. So the father's traits are passed to sons only. So here, dominance is actually irrelevant because there's only one copy of each Y-linked gene. This is actually known as hemizygous. And as you can see here with another example, males only passing into the male, male passing into the male, same thing here, same thing here. All right, let's try a few questions together. Hopefully you have your cheat sheet out. Definitely use that to ensure that you can get the correct answers, all right? All right, the question is going to be, is this autosomal or is this X-linked? And I'm not asking you to tell me if it's dominant or recessive, just tell me, is it autosomal or is it X-linked? Pause the video, give this a try on your own, resume when you're ready. Alright, so let's try the checklist. Alright, you should have guessed it, it is definitely autosomal. It is an exact 50-50. Alright, we got 1, 2, 3 males that are affected, and then we have 1, 2, 3 females that are affected. Alright, now I want you to figure out, is this dominant? Alright, or is this recessive? Okay, you don't need to tell me if it's autosomal or X-linked, just tell me one of these two. Give it a try, pause the video, and then resume when you're ready. Alright, it is definitely dominant, okay? All affected individuals have an affected parent. Affected with an affected parent. Affected with an affected parent. Alright, let's do one more before we start putting it all together. Again, is this dominant or is this recessive? Pause the video, give it a try. Alright, so this is obviously recessive. All affected individuals have an affected parent. Is that true? No. This affected individual doesn't have affected parents. This affected individual doesn't have affected parents either. So it is definitely recessive. Alright, so this one, question is, what is the pattern of inheritance? So this is obviously everything. Tell me, is it dominant or recessive? And X-linked or autosomal? Pause the video, give it a try on your own. Alright, all affected individuals have an affected parent. This one, yep, affected parent, so this is definitely dominant. All affected males have affected moms. Male affected mom, that seems to be true. Is it X-linked? Maybe. All affected fathers have affected daughters. All affected fathers have an affected... Oh, no, this is not. It's not an affected daughter. So, maybe is it not X-linked? We'll do one more. All affected fathers have no affected sons. That's obviously not true. The son here is affected. So it's definitely not X-linked. This is autosomal. Alright, let's try this one. Pattern of inheritance. Give it a shot. Pause the video, try it yourself. All affected individuals have an affected parent. Nope, that's not true. It's not dominant, so this has to be recessive. Alright, is it mostly males or is it mostly females? As you can see, it's 50-50. So, it's likely going to be autosomal. However, because the sample size here is very small, let's see if it actually works out if we are categorizing this as recessive autosomal. If it doesn't work out based on our categorization over here, then this is wrong. Then this is probably going to be X-linked. Let's try. So if this is autosomal, it's not XX's or XY's involved, right? It's just big R, small r's, so let's try those. So let's have our little Punnett square. Obviously, if it's recessive, that means that it is going to be the small r's that are going to be the affected trait. Alright, so that's the dangerous one, the small r's. So this one over here has to be a small r. So the big R's are going to be the dominant ones, right? So they're the healthy ones. So I'll just put here healthy. Or normal. Obviously this one and this one are normal. So is this going to be a big R, and a big R, or is it going to be big R, small r, or is it going to be a big R, small r, big R, small r? It has to be this, because if you were to do it this way, look what happens. Big R for the male, let's say big R, small r for the female. You get big R, small r, big R, big R, big R, small r, big R, small r. All of them ended up being normal, quote unquote normal. None of it leads to a chance of possibility of having a small r, so has to be heterozygous for both parents. Alright, so let's just change that to a small r over there. So you get big R, big R, big R, small r, big R, small r, small r, small r. So what does this one have to be? This one can be either big R, or big R, small r. So I'll just put a big R with a dash. So it works out to be recessive autosomal, so you'll be awarded all points because it makes sense genetically, based on the categorization that you put at the beginning. Alright, same thing. Tell me the pattern of inheritance. Give it a try, resume when you're done. Alright, let's do the checklist. All affected individuals have an affected parent. Individual affected, affected parent. Individuals affected, affected parent. So this is probably going to be dominant. Alright, is this going to be autosomal or is this going to be X-linked? Remember, if it's X-linked, all affected males have affected moms. Affected male, affected mom. Okay, so this could be X-linked maybe. All affected fathers have affected daughters. All affected fathers have affected daughters. Alright, so we're getting even closer. All affected fathers have no affected sons. This is definitely going to be X-linked for sure. And if you want to be extremely careful, start putting in your alleles, your pair of alleles for every single one of these, and ensure that it makes sense for it to be a dominant X-linked. Being meticulous in this regard will absolutely help to ensure that you make no mistakes on your assessment. Alright, try these questions on your own. They are down here, okay, number 1 and number 2. Obviously use this as a reference. Pause the video, resume when you're ready. Alright, so in this question over here, and the interesting is they actually tell you that red-green color blindness is what we're looking at. That's the disorder here, and it is X-linked recessive. And this is very important because, again, these are just a snapshot from a much, much larger family tree. So if you did your checklist and it turns out to be autosomal, that's because the checklist is not foolproof. Okay, it's not big enough. This is just a small snapshot. So in this case, they tell us that it's an X-linked recessive trait. So again, recessive is the disorder, right? So obviously a small r has to be the disorder. So this is going to be the affected one. All right, and the big R is going to be normal. So let's do our Punnett square and cross what we need to cross. Again, it is X-linked, so we got to start doing the X-X's and the X-Y's. All right, so let's put the X-Y up here. Let's put the X-X here. So let's look at the affected one first, okay, because that's the important one. Okay, which one has this disorder? So we got to look for the one that is shaded. All right, so let's look at this one first. This one is shaded. So this is the female. So the female, if it's shaded, it means that she has to be homozygous for the small r. So it has to be a small r here and a small r over there. What does the male have to be? The male has to be healthy, so it has to be a big R. So I have to put a big R over there. So we get X to the big R, X to the small R, X to the small R, Y, X to the big R, X to the small r, X to the small r, Y. So let's look at the offspring over here. Actually, let me quickly label this. So this is the female, so this is X to the small r, X to the small r. This one is X to the big R, Y. And again, let's look at the affected individuals. Okay, so affected males. Okay, which ones are affected males? Yeah, it makes sense because 100% of them, males here, are going to be affected. All right, so this is for sure X to the small r, Y, X to the small r, Y. Remember, all affected males have affected moms. Or let's look at the female over here. The female has to be, again, 100% heterozygous genotypically. So this is X to the big R, X to the small r. All right, let's try this side over here. Another Punnett square. Let's cross them. So this offspring, just one female, and is normal, okay? So let's put the parents up here. The male over here is affected, okay? That's what we always going to look for, the shaded ones first. So we're going to put an X to the smaller because that's the only way that it can phenotypically show that the male is affected, right? So how about the female, okay? If we did female big R, what's going to happen? You get X to the big R, X to the small r, X to the big R, Y. Then you get X to the big R, X to the small r, X to the big R, Y. What happened to the female? Female here is X to the big R is healthy. And that's fine based on what we looked at. It could be X to the big R, X to the small r. All right, so it could be X to the big R, X to the small r. And obviously this one has to be X to the big R, Y, right? Want to try another one? Instead of this being x to the big R, x to the big R, like we did over here. Do you want to try x to the small r? Okay, does that work? Let's try it over here. All right, so you get this one, x to the big R, x to the small r. This one, x to the small r, x to the small r. And this one, x to the small r, y. All right, it works out too, right? Look, well, it obviously can't be this one, right? Because this one over here would mean that it is affected, the female, right? But this female is not. The female is showing a normal case of a phenotype. So it has to be this one, x to the big R, x to the small r. So either one could work, actually. So this one could be x to the big R, x to the small r, or the previous one we did, x to the big R and x to the big R. So I'm going to be putting here that one, okay? A dash. All right, and what does this one have to be? Let's do another Punnett square. I know we're doing every single one. The questions aren't really asking for it, but it's always good to do everything. It is always best to go above and beyond and make sure you make no mistakes anywhere. Just do everything. So again, xy, xx. So what is the male here? X to the small r, y. And this female, x to the big R, x to the small r. So you get x to the big R, x to the small r, x to the big R, y. X to the small r, x to the small r, x to the small r, y. So again, this is the female, right? So it could be this female or this female chance. Obviously, you can only use this one because this is the only normal phenotype. This one is going to be affected. And as you can see over here, it's not shaded in. So it has to be that one. So this is x to the big R, x to the small r. So let's answer these questions. What is the genotype of 2, 1? All right, so this is row 2 and number 1 over here. All right, so this would be x to the big R, x to the small r, right? All right, and then 2, 3. All right, so same row 2 for the third one. This is x to the small r, y. So x to the small r, y. All right, what is the genotype for 3, 1? That's this one we figured out over here, right? It's x. It's heterozygous. X to the big R, x to the small r. All right, let's put this over here. X to the big R, x to the small r. All right, what is the probability that 3, 1, so the one that we just looked at, will be colorblind? Well, we're only dealing with the female, right? We know that the sex is female, so it has to be between these two. So there is a 1 out of 2 chance being colorblind. So a 50% chance, all right? So 50% chance. And in this case, luckily, the female ended up not being colorblind. She ended up being this one over here, not this one. So she's normal. All right, this is going to be our last question. I really want you to try this on your own if you haven't been trying these on your own by yourself. This is very important. So we have a final question over here. You have this pedigree chart to label, okay? Obviously, you know what I'm going to be asking you for. What is the pattern of inheritance, okay? As well as guessing the genotypes of every single person over here. So what are the genotypes for every single individual? All right, and I don't mean single as in like not married over here. I just mean like every single one. Like make sure you get the genotypes of every single one over here. Okay, so the pair of alleles for every single person. And obviously you need to make sure that you know the pattern of inheritance that I just said about. Because without the pattern of inheritance being guessed correctly, you won't be able to get the genotypes correctly. I assure you that. So try this on your own, pause the video, and only click resume when you have done it yourself. All right, so this here is the answer. Hopefully you got it right. And it is, again, it is very imperative that you know whether it's dominant or recessive, right? So this is 100% dominant, okay? And this is also 100% autosomal. As you can see with the pairings of alleles, right? You don't see any XXs or XYs with those letters on top. And I actually want you to try to do this with an X-linked pathway, okay? So just put like XYs and like XXs and give it a try. And I assure you, when you do this in an X-linked fashion, you are going to get stuck. 100% stuck, because it's just not going to physically make sense in terms of the chances of probability. It will not work if you do it X-linked. So if you guessed X-linked by accident and you actually implemented it and you actually answered your pairings of alleles in that fashion, in that pathway, and you get stuck, that is a good indicator that you made a mistake. So you would have to erase those and then try it from an autosomal lens and continue adding your pairings of alleles in. And I assure you, you won't hit that barrier again, because it's going to make sense statistically. And then you can continue to the very end of your pedigree chart. And this is what you should be getting for your answer.